Hello, everyone, and welcome to today's webinar. First, let me introduce myself. My name is Stefania Cesarano from Nemis Technologies. I'll be responsible for hosting this webinar today. Maybe let's wait for another 1-2 minutes. In the meantime, I will briefly explain the program. After introducing today's speaker, Lukáš, he will take us into the wonderful and amazing world of bacteriophages. Anyone who knows me knows that I love bacteria, but with bacteriophages, we are going a little bit deeper into the microcosm. We are all here because we are part of the food safety community, and yes, phages are also part of food safety. So please interact and be a part of the webinar and submit your questions in the chat room. After the presentation, there will be a Q&A session. I will try to get to all of them on this webinar so that Lukáš can answer them. If I do not get to all your questions, as we have limited time, there will be a Q&A on the Nemis Academy website. Great. I think, Lukáš, you are ready?
Perfect.
Okay, our topic today is bacteriophages, a new opportunity in food safety. I'm sure we will learn a lot from Lukáš on that topic. Please, Lukáš, introduce yourself.
Thank you very much. Thank you very much, Stefania. Welcome, everyone, on this webinar. As Stefania was kind to introduce, I'm Łukasz Richter. I am a product manager at Nemis Technologies. I am a huge fan of bacteriophages. I'm actually a microbiologist and biotechnologist. I did my PhD thesis in bacteria detection using bacteriophages, and I work with them for over 8 years now. I'm absolutely fascinated by them, and I hope by the end of this talk I will convince you that they are amazing little entities, definitely worth keeping an eye on, also in food safety applications. I think I can share my screen now, right? Right. I hope you can see the screen.
Yes.
I'll just hide the thing. Right. So today we're going to talk a little bit about bacteriophages. We're going to start with what are they and basically what do they do, where we can find them. We're going to delve a little bit into the world of where we can apply bacteriophages or where humanity so far applied them. We're going to focus especially on something called biocontrol, bacteria detection, also in food. And at the end, there will be a little bit of Q&A section to answer all your questions. All right. So let's start with what are bacteriophages? In short, bacteriophages or phages are viruses that infect bacteria and bacteria alone. So like some other viruses that you may know for humans, like coronavirus, for plants, for pathogens, for plants, these viruses infect bacteria and bacteria alone. And it is estimated that there is around 10 to 31 phages on our planet, which is a rather abstract number. But just to give you a good understanding, there's more phages than any other organisms combined, including microorganisms and bacteria. So they are the most abundant entity on our planet. They can be found literally everywhere. All the ecosystems contain a part that is bacteriophages, in water, in soil, in air. So they're extremely important piece of modulating or of controlling the whole ecosystems and the whole microbiological walls of the ecosystems everywhere on the earth. Even as humans, we have phages in our body. Again, it is estimated that we have around 10 to 13 phages in our body. This abstract number can be a little bit better described by telling you that there are more phages in our human body than bacteria in our guts, in our skin. So we have more phages in our body, even in our own cells. And despite their abundance, despite the fact that they're everywhere, we only know them for a little bit more than 100 years. They were discovered by Felix Derel in 1917. And one interesting, I think, fact about bacteriophages is that there was even a Nobel Prize In 1969, three people got the Nobel Prize when they discovered how these little fascinating creatures multiply, how they live, and what's their life cycle. We're going to delve into this a little bit more in a second. So on the right, you see the structure of bacteriophage, or the build of the bacteriophage as it's typically presented. And now you may ask, but is that how all the bacteriophages look like? And the answer
Excuse
is,
me,
right,
Lucas, just a second.
yes.
Can you hear? Can you ask the other people if they hear you?
Yes. Do other people can hear me? Some say yes, they do.
Okay, cool.
All right, great.
Super. Thank you very much. Sorry, Lucas.
No problem, no problem. Right. So about how phages look like, it is true that most phages share this tailed structure or head-tail structure as I presented originally, but not all of them are like that. We have phages that have this head, which we call capsid, tail, and these little legs, which we call fibers, but this tail can be long, can be short, can be non-existing, it can be rigid, it can be flexible. The phage can be just basically a protein ball with membrane or without, or it can be even this long, elongated structure looking more like an earthworm, microscopic earthworm, than any other thing. And phages took these different structures, different forms, and as you can see here, also different types of genetic material they store to tackle the problem of infecting so many different bacteria. They evolve into different forms because they need to evolve to attacking all kinds of different bacteria that are existing in our world. And this abundance of forms and shapes is both boon and a bane. We can use them in different applications, but we also have to take into consideration that every phage will be slightly different. Now, okay, we know how do they look like, but now the question is, what do they do? What is the normal life cycle of the bacteriophage in the wild? So let's assume that we start with three phages just floating in the ocean. The first step is that they use the end of the tail and fibers to very selectively find the bacteria that they infect. So this process is very selective. It helps phages to anchor at the surface of the bacteria. They drill a little hole in the cell membrane, and then they inject, like a little syringe, the DNA into the bacteria. And then the hijack starts. But bacteriophage basically hijacks the whole bacteria, forces it, instead of doing its normal, regular things, to start replicating and creating new virons, new bacteriophages inside. When the time is, when everything is ready and it's time for phage to be released, there is a signal for the bacteria to basically kill itself, to destroy itself, and new fully formed phages, which we sometimes call progeny phages, are released, and the whole cycle can close. This cycle is typically called lytic because we have lysis, so killing of bacteria, and this cycle is most commonly used in practical applications because we want phages to kill our bacteria, and in this cycle, they're for sure killed. But that's not the only option. There is one more side option for the bacteriophage. After it enters the bacterial cell, after the DNA is injected, it can also hide in the bacteria. It can build its DNA into the bacterial DNA and stay in this hidden form for many, many generations of bacteria. Bacteria can grow and multiply and create new bacteria with this hidden form of bacteriophage, which we sometimes call prophage, embedded in it. And when it's time, when the phage is ready or it's decided, there are special molecular mechanisms that the phage can use. It can do an induction, and the phage can switch to its typical lytic killing cycle. So phages are for sure fascinating, but why we think they're useful for us human beings? Why we thought when we looked at them, when we studied them at the very beginning, that they're so great and we definitely need to use them? Well, first of all, they're highly specific. So typically, a given type of bacteriophage will infect only single species of bacteria. Sure, some of them are broader, few species of bacteria, some of them are narrower, for instance, only a subsection of strains of a given species, but we can find phages that are covering all these different ranges for basically all the bacteria we know. So we can very finely tune the type of bacteriophages or mixture of bacteriophages we want for a given job. they are considered to be green technology, right? They come from nature, we isolate them from nature, so they're green, they're biocompatible, they're completely harmless for humans, animals, and plants, and they have a status GRAS, which means generally recognized as safe. Phages also are famous for their high potency, which means that even a small dosage of phages, even a few phages, can start the process of killing bacteria and very efficiently wipe out even the whole culture. And this is sometimes connected, or it is actually connected to the fact that phages do a self-replication during a cell cycle, right? So you can imagine that we have phages smeared all over the place, but there is a hotspot of listeria, a culture of listeria sitting somewhere. Phages will start infecting bacteria there, multiplying, and the concentration of bacteriophages locally increases. So it's like a self-replicating drug. It is also proven that they do not affect organoleptic properties of food, which is, of course, especially important in food safety. Phages are basically a bunch of proteins and a bunch of DNA assembled in the proper way. And the amount of phages that has to be applied because of their high potency are typically not high enough to influence any organoleptic properties of the food they're sprayed on. Phages are also known to be effective against biofilms. They can diffuse deeply into the biofilm layers and reach bacteria typically protected with these layers of polymers that are building biofilms. Last but not the least, they're also effective against multi-drug resistant bacteria. And it's because any mechanism that the bacteria develop for any drug resistance is completely independent with a mechanism of phage infection. So even if bacteria can resist any given drug, it's completely not influencing what the phage can do while infecting the bacteria. That's why it's so great. It's an alternative. All right, so phages are great, but is there really a need for humanity? One rather obvious thing is that wherever we have bacteria that are causing a problem for us, phages looks like a good option, right? We are using antibiotic for so long. We partially because they're so common, also because not everybody is using them correctly, we created a lot of drug-resistant bacteria, which are now started to causing more and more problems. We don't have new antibiotics, so the amount of deaths caused by these drug-resistant bacteria is really high and will only increase according to estimations. So one thing is that maybe let's stop using so many antibiotics and look how phages can help us when we do antibiotic therapy. And second, rather obvious and actually very early idea was usage of phages in livestock. Of course, we also feed our livestock different antimicrobials, and the amount of antimicrobials a given animal takes throughout its life before it's being slaughtered is really high. And these antibiotics gonna partially stay in the body of the animal, so later it will end up in our food. So the idea was that why not start using phages anywhere when there is a livestock? And later it was even, of course, transferred to farming, but we're gonna touch on that a bit later. So now we know that there is a need. Phages look like a great option. And now I would like to show you a little bit, a broad overview where phages are applied to date. What are the most common applications of phages? Not only in food, but in general, how we learned as humanity to use them. The very first application is called phage therapy. This was the very first idea the moment they were discovered, and basically the concept is simple. Whenever we have bacterial disease in humans, let's treat these bacteria with bacteriophages, right? It's rather popular, especially these days when it has its renaissance a little bit. I'm going to describe in a moment why, but that's definitely one of the most common usages of bacteriophages these days. Second application or second method that is using bacteriophages is called phage display. It's a little bit less known, but to give you a short description what it is, it's extremely important tool in drug development. So if we want to find a way how we gonna create a molecule that will go into our kidney, because we want to deliver a drug directly into our kidney, this method, phage display, will allow us to screen millions of molecules just to find this one particular that will help get our drug directly to the kidney. And this technique was proven to be important enough that there was a Nobel Prize, as you see here in 2018, in the field of drug development. Of course, we have problems always when we produce food in livestock, in farming with bacteria. So people started applying phages in these applications. When we harvest something or when we slaughter animal, of course, we switch to food production or bioprocessing. And there also, of course, there is a big problem with bacteria, not only in food facilities, so environmental monitoring, but also we want to do the food preservation to stop bacteria from growing during shelf life. And everywhere, phages, people had ideas, let's apply phages, let's try to do something like that. These two things together, so a little bit from farm to fork, is broadly known as biocontrol. So that's a general name for this application. And the last one that is rather popular is bacteria detection, because phages are so selective, right? They have this tail and these fibers, and they can selectively find a given bacteria. So why not use this property in the field of bacteria detection? Right. So before we switch to food, I want to also tell you a little bit what's our relationship with bacteriophages as humanity, because we had our ups and downs. And the example will be phage therapy, which is I think, the best example because it's a leading use of bacteriophages. And here you have a timeline, basically, of recent human history, and then on the y-axis you have a qualitative interest, or how much interest we put, how much research we did, how much scientific papers we produced for a given time. So of course, the moment we discovered bacteriophages, the interest was super high. Everybody was extremely enthusiastic. We have something against bacteria for the first time. 2 years after the discovery, we had a first phage therapy for a disease called dysentery, and everybody was really interested in that. But then, 12 years later, something happened. Alexander Fleming discovered the first antibiotic, penicillin. That matched in time first critical voices against phage therapies because it was not reproducible, it was not working that broadly. And at that time, our knowledge of genomes, molecular biology, protein-protein interactions was rather small. So we couldn't control phages that well. We didn't understand them. So phages went a little bit away, especially after introduction of penicillin in treatment in 1942. Bacteriophages were not that interesting anymore, and antibiotics were everything. During Cold War in between 40s and 70s, this time it's sometimes called golden era of antibiotics. New antibiotics were discovered. They were broadly used everywhere, spread from original uses like military all the way to homes of ordinary people. And the only place where bacteriophages were still studied, because of course, America and the old Europe, they had antibiotics. So the former Soviet Union wanted to have something else, not like the rotten West. They wanted something on their own. So phages were still studied in former Soviet Union, and phage therapies were still developed there. And we see the effect until now. Still, the best places for phage therapy in the world are in countries of former Soviet Union, in Georgia, in Russia, in Poland, and so on. And only recently, only 50 years ago, we started getting tools that allowed us to understand bacteriophages better, and we started developing new, way more controlled applications of them. We sequenced the genome for the first time. We've proven that phage therapy is actually working against antibiotic-resistant bacteria, which was back then was proven to be actually a problem. And then in 2006 only, there was the first commercial phage product for biocontrol produced by a company called Intralytics from the United States. And only 9 years ago was the first clinical trial for phage therapy. The first clinical trial, I have to say, according to good clinical practice, as sometimes people describe it, was done in European Union. It was called phagoburn. But even that one was claimed to have a mixed success at best. But now phages have the renaissance. We don't have antibiotics anymore. The ones that we use more and more often are creating antibiotic resistance in bacteria. We create these so-called superbugs, but we understand phages better and better. So now it's the renaissance. Now is their time. All right, but let's switch to using phages in biocontrol them, right? They were great in phage therapy, but we can also apply them in our food production chains. And I looked at different applications that phages phages have in biocontrol, and it's actually extremely broad, from scientific publications to some first field studies, all the way to commercial products. To just show you an overview, phages were basically applied anywhere where we found problems with bacteria. So if you think about the first stage of production of food, so livestock, farming, and crops, you can see that every type of such an activity, from livestock to fish, seafood, vegetables, fruits, everything, we had an attempt at least to kill bacteria with bacteriophages. And even here in Switzerland, even here in Waddensville, we actually have experts in this field. As NEMIS, we have a pleasure to work with Professor Lars Fiesler from ZAW, and his group was involved in the project called Phage Fire, when they were finding phages against Erwinia, that's a bacteria that is causing fire blight in plants. So, you know, the expertise is also just next to us. Then when we go to food processing or food bioprocessing, of course, the most dangerous bacteria there are typically Salmonella, Listeria, Escherichia coli, and then Campylobacter and some other. And of course, there in every segment, there are publications, and the publications are actually scientific publications released every day, that in every segment we can find at least an attempt or a described scientific successful attempt to kill bacteria with bacteriophages. And as a humanity, we even went as far as we are using phages in wastewater bioremediation. So we even use phages to deal with our waste. So now you can ask me, okay, scientific publications, great, but is it even available? Do people use it? Can people buy it? And yes, there are commercially available products. And when I did the research and I looked at different companies that provide phage-based products for biocontrol, these three were the most prominent. The one in the middle, Intralytics from the United States, is by far a pioneering company. They had the very first application. They were very quickly followed by Micrios, company originally headquartered in Netherlands, still headquartered in Netherlands. They even have a little division here in Switzerland, in Waddensville, actually. And a little bit less known company, it's the company called Brim Medical. It's a bit less known because it's from Georgia, and despite the fact that they have a really broad portfolio of their products, they're not exactly following all the requirements of the Western world. But these are not the only three companies that are doing it. As you see here, there are actually quite a lot of companies all over the world doing their own attempts to provide phage-based product. Each of these companies, as you see from United States, from different places in Germany, from different places of Europe, these companies have their own offer for customers, their own know-how about types of phages they use, formulations of phages they use to provide the best effect. So you see all these products, and as you can see here, a lot of these products are for Listeria, so of course Listec, List Shield, and Mono are of course for Listeria monocytogenes, S or Salmonella are for, or Salmo are for Salmonella, and Escherichia coli. So we can ask now, okay, there is a product, but how does it look like? Is it powder? Is it how can I use it in my food factory? One thing I want to mention before that is that Intralytics and Micrios are the most prominent companies, not only because they were first, but also these are the only two companies, to my knowledge, to my research, that were approved by FDA, and Micrios has even endorsement from EFSA. There is a document of EFSA describing usage of Listex P100 that it's allowed to be used in food factories. So now we can ask, how can we use these products in practice? How do they look like in practice? So to start with, by far, most products will be in one of these three applications. It will be either Salmonella, either Escherichia coli 0157, or Listeria monocytogenes, right? And then they typically come as a liquid that can be sprayed directly on food. The segments are really different. Each company has their own description of the offer, but typically it's divided into meat and poultry, fish and seafood, vegetables, and there is even dairy. Some companies offer things for dairy segment. And the general procedure is that there has to be a cleanse in the process in the process of production somewhere on the conveyor belt, maybe when you spray solution of phages directly on your food, or you spray the solution of phages in your factory in the places when you would like to get rid of given bacteria, for instance, Listeria monocytogenes, but maybe without using disinfectants, or maybe after using disinfectants to be even more precise. So that's typically how without getting into the details of every product, how it typically looks like for practical application of phage-based products commercially available for biocontrol. But now you can ask, all right, it looks really great, but why phages are then not so broadly used? Why they didn't kick out completely antibiotics and cleaning agents and everything? Well, it's because there are still few challenges we need to address, like with any new-ish technology, because I just told you phages have their renaissance actually, and there are still things that are need to be addressed before we can spread the usage of the products everywhere. One of these bottlenecks is a large-scale production and purification of phages. And don't get me wrong, there are methods very well established to produce some of the phages. But because I told you at the very beginning, phages have so different structures, shapes, and forms, there is a need for a universal method that will work for all the phages to produce them at big scale and purify them properly at the big scale. Or maybe there is more research needed to find these different methods for all the phages we know. Then we still need to work on phage stability. The phages are typically exposed to many different conditions, right? With pH, temperature, humidity, stability in time. And every phage will react slightly differently. And it's actually an art, it's a know-how, how to create a given phage formulation, what to add to our phage products so that they retain their stability in all these different conditions. There's also always a question what's about the resistance to chemicals, for instance, cleaning agents. We have to make sure that they will work hand in hand, because very often, especially for cleaning purposes and stuff like that, they will be exposed to the same cleaning agents that bacteria are in our food factory. Sometimes phages are also responsible for releasing pro-inflammatory compounds. This is more for phage therapy, where when we kill bacteria and we lyse them open, we cut them open, whatever was inside spills out. And some of these compounds can be considered pro-inflammatory. This is less of a problem in food factories, where the concentration of these compounds are basically minuscule, but it's a little bit bigger problem that is still being addressed for phage therapies. Sometimes we also find out that phages have too narrow host range. I told you that they have extremely well-defined host range, and that's absolutely true. But maybe realizing after doing some field studies, or after finding phage that has amazing properties, that its host range is actually not the host species as we were hoping, but only a group of substrates. And some bacteria we encounter are then resistant to this phage. That's also sometimes a problem. So this precise definition of the host range of the phage, so which bacteria can a given phage really kill, is also something that has to be addressed every time. And finally, in rare cases, bacteriophages can coat toxins like Shigella toxin. It's really a case for the few phages, but when we design or when we think about applications of phages, it also has to be checked if the phage we're using is free from these things. Luckily, these days it's rather easy to do, but that's one of the things that is also out there. Right. So we said that phages can be used in biocontrol, and that's all great. And the environment of using bacteriophages in biocontrol is so homogeneous, right? So maybe we can use bacteriophages somewhere else when we can control the environment better, and therefore we can, a little bit more precisely design our application. So phages, as I said in one of the previous slides, can be also used in bacteria detection. They can find bacteria very precisely, and actually we're using that fact that phages will infect or find or attach to a given type of bacteria in bacteria detection. Just to give you an overview, there are many types or ways how we can use bacteriophages in bacteria detection. I was the part of that field when I was doing my PhD thesis, And phages can be applied, as you see here, on the surface, and we have sensing layers. We can infect bacteria and then use that fact to detect something that changed in the bacteria, or we can use them as capture element. And despite all the effort, despite having so many publications out there, to date, the only commercial application or commercial product that was available that uses bacteriophages that are directly infecting the bacteria in the device was a device called Sample 6. This device was using phages to detect Listeria spp. And that product is no longer available. It's vanishing from the market, is I think rather mysterious, but it only shows that the field still needs to mature. It still needs more effort and understanding to find a first proper commercial application, or now it will be second commercial application, to use phages directly in bacterial detection. But if we cannot use them to directly detect bacteria, maybe we can use them in one last way I want to show you today. Phages, as I told you, are extremely good killers of a given bacteria. So why not use them in our enrichment broths to improve selectivity? When we enrich bacteria, typically we enrich bacteria when we want to find them. So when we do a detection step, we care about selectivity. We only want to support the growth of our pathogenic bacteria. So we can, and we have a bunch of different agents we can apply to support our target bacteria and to inhibit all the other bacteria that we don't want to grow in our broth. So we can think about it like that. When we start, it's like a funnel. When we start, there's a lot of bacteria in our sample. And only one of them, or maybe only few of them, is our target bacteria, which in this case can be Listeria monocytogenes. Then we add antibiotics. Antibiotics will wipe out some of the bacteria. Some red, some violet, some orange are gone. Then we can add some salts. Lithium salts or bile salts, and again, few more bacteria are inhibited. And then finally, what we can do typically is apply proper temperature that will only promote growth of our pathogen. But even then, still few bacteria may sneak in. Maybe they're too similar to our pathogen and they will look like false positive, even though it's not our pathogen. Or maybe, on the other hand, they're extremely good competitors, surviving everything else, and they will outgrow Listeria. And that's false negative. So what we can do here with bacteriophages, we can design or add bacteriophages designed specifically to target these few problematic bacteria. And now, because we can finely tune which bacteria we're gonna target, we can wipe out these few remaining bacteria, and our broth has proper full selectivity. And you may ask, okay, is someone using it? Is that approach even proven to be effective in a practical application? And my answer is yes. At Nemis Technologies, we are actually using pioneering phage technology to improve selectivity of our broths. We have this little tube test, this rapid lab-free pathogen testing method, where we use a single tube, and in that tube there is enrichment broth that contains special mixtures of bacteriophages designed to eliminate these few problematic bacteria we couldn't remove with all the other selective agents. So this is like an extra layer, an extra level of selectivity, which we provide in our tests. So if you want to see phages working in real life, hands-on, I definitely encourage to try them. That was all I wanted to tell you about bacteriophages for today. Thank you very much for your attention, and I'm open for any questions.
Thank you very much, Lucas. Amazing and really wonderful world of phages. Let's see if there is one good question. Phages are sensitive towards strains, not species. How can this limitation be overcome or even used, Lucas?
Right. So phages are, they have a given host range, right? And it can happen that their host range is broad enough that the number of strains covers the whole species, right? In very rare instances, typically we say that if there's 90 to 95% coverage of a given species and only few strains we found that are resistant, we consider this the phage that is covering a full species. it will almost never be perfect because bacteria are fighting back. Bacteria is always fighting back for whatever you throw at them. So of course we can always find this one strain or these few strains that will be resistant, but broadly speaking, we can find phages of broad enough host range to call it the whole species. How can we use it, or how can we overcome it? Of course, we can keep on looking. We're finding new phages, cataloging new phages every day. There are places with extremely broad libraries of phages to check and select from. We need to keep on characterizing them. And the new attitude that is still very scientific, I think it's not used in practical usage. People are learning how to modify phages, how to learn from everything we know so far to design from scratch a perfect predator, a perfect phage that will kill exactly what we want. So that's a little bit my answer on that, my take on that that case.
Thank you. There is another question. Oh, how can we get clear approval for use of, for example, Bacteriophages or products of animal origin? Should codex be engaged with?
Right. So here, I don't know all the details about laws in different countries, how phage products can be applied. As far as I know, European Union more or less unified its take, but I don't want to mislead anyone. I think it has to be checked in a country by country base. And honestly, I was never deep enough inside into the usage of phage-based products in animal origin or in animal related food facilities to answer that question, in full. So it just has to be checked for a given country and maybe consulted with a body responsible for this, like for European Union itself.
Okay, there is another one. We still have time. Yes. Antibiotics often also affect, to a certain degree, the target organism that should be enriched. Do phages have a similar problem?
So right, we're talking about phages that may by accident target our pathogen we would like to enrich. So it's always checked, right? So if we design a method where we want to grow a given pathogen and we select our phages that we would like to use to kill everything else, it's checked. At Nemis, for instance, we just checked all the phages if they're infect, if we use it for Listeria monocytogenes, if they infect Listeria monocytogenes, as many strains as we can. And then we're sure that the phage is not influencing our pathogen. And in a lot of cases, it is the case that the phage will not touch our pathogen because of their very well-defined species selective, even strain selective host range. So that's very rarely the case. I think it's more common to have a contamination of some other phages because of bad production quality or maybe the processes, rather than having a phage all of the sudden infecting our pathogen.
There is enough. Thank you. There's another one. Oh, amazing work. Congratulations. Are there some tech that may overcome the lack of motility, random interaction with the hosts for food application?
I'm not sure what lack of motility means in this context. Random interactions with the host. Yeah, I'm not sure if I get the question. I don't know if the person that wrote this question can
yes, there's someone typing.
Explain a bit more for this question. Yeah.
Maybe in the meantime, I have also a question. Yes, okay. Oh, no. No, it's not this one. Okay. Maybe waiting for more explanation. One question that's myself also, I'm thinking often of it. You told us about external factors that can impact the effectiveness of phages, right? thinking of our system, phages can be in some kind of osmotic stress. If you are swabbing some kind of extremely saline with a lot of salt, and we put the swab into our enrichment with the whole phages cocktail in it, can it possible that in that time the phages come in some kind of a stress and they won't do their work?
Mhm. So you touched on the very broad problem, which is phage stability. And phages don't feel stress or osmotic stress like bacteria do, right? Because bacteria have a balance between what's inside them and what's outside them. Every cell does. But phages are much simpler. They're just a bunch of proteins assembled properly with a bit of DNA in their head. So phages can be deactivated in given conditions, but it's always about checking it beforehand. And there are phages extremely stable or extremely resistant to, for instance, high concentrations of salts or different temperatures or anything that can cause osmotic stress or any other property of the environment. So there are phages that can deal with a given things extremely well, but we also know phages that are extremely weak. And it's all about finding the phage that will work correctly in a given application. So we should always define what is the range of given parameters. So if I'm spraying it in, I don't know, food factory, in dairy factory, there will be humidity, there will be temperature, and my phage has to work from 4 degrees, or ideally even like 2 degrees, all the way to 30 degrees, right? And I need to find phages that will do this for me, because if not, they will be deactivated.
Oh, okay, cool, thanks. Well, there are other questions. Lukasz, I think we have time for another one. It is related to the spray on top of food.
Right.
How to encourage the contact between phages and host?
Right. The answer is you cannot. Which means you have to spray enough phages to make sure that they're gonna eventually meet proper bacteria or bacteria we want to kill. So phages will move. They will move in the environment. They will move in wet environment via diffusion. So this normal random motion that every molecule, every atom, every entity in the microworld has. Bacteria will move because of their motility. They will move because that's what bacteria do. And that's how phages work. And by random chance, phages will find the proper bacteria, will actually expose their little fibers, like little legs, to find that proper bacteria. So it's all about spraying them evenly on your product and making sure that the concentration is high and that the phages are not deactivated in the conditions that you're applying them in.
Great, you're really enthusiastic about this.
Always.
Always great. We have not much time anymore, I think. Just for me, as kind of a thank you very much, Lukasz. Very thrilling. I'm also like this, but with bacteria, not with phages. It's clear that phages represent a groundbreaking tool in food safety. By leveraging their power to detect and fight harmful bacteria, we are not just advancing technology, but leveraging how we ensure the safety of our food supply. Now, join us for our next webinar on the 27th of February as we delve into unveiling food safety excellence beyond age HACCP. Discover how holistic approach integrating various practices, standards, and systems elevates food safety. Join on the upper panel button, please. Register this red button up, and then thank you for being a part of this journey and a part of this community. Have a nice day.
Thank you very much.
Thank you, Lukasz.